T2 bacteria is a virus that specifically parasitizes E. coli. Its head and tail shells are made of protein, and the head contains DNA. After infecting E. coli, T2 bacteria will use the material in the host bacteria to proliferate in large quantities under the action of genetic material until the E. coli is lysed and the bacteria are released. In 1952, American geneticists Hersey and Chase used T2 bacteria as experimental materials to find out whether the genetic material is protein or DNA. The whole experiment is very simple. They only need to use 2 radioactive substances to mark the bacteria and then let them infect E. coli. Finally, they can analyze which substance invaded the bacteria. First, they cultured E. coli in culture media containing radioactive isotopes sulfur 35 and 032, and then used these E. coli to culture bacteria. In this way, they can obtain bacteria with sulfur 35 markers in protein and 032 markers in DNA. Then they are allowed to infect the labeled E. coli. After a short period of incubation, they are stirred and centrifuged in a mixer. The purpose of stirring is to separate the bacteria adsorbed on the bacteria from the bacteria. The purpose of centrifugation is to remove the lighter bacterial particles from the supernatant, while the heavier precipitate is E. coli. After centrifugation, it was found that a group of experiments using sulfur 35 labeling
were mainly distributed in the supernatant, and a group of experimental radioactive isotopes labeled with phosphorus 32 were mainly distributed in the supernatant.